All right, last class we set the stage with a lot of new chapter 4 vocabulary. But now in 4.2, we're going to switch to a very specific type, and that's the idea of what is an experiment. So last section, we learned that an observational study observes individuals and measures variables of interest, but does not influence responses at all. So sometimes people get a little confused on what falls under observational study. An observational study is almost anything. It is looking at past data. It is looking at artifacts. It's asking people questions. It's researching some sort of really kind of researching anything that happened already in the past, researching, but it is not imposing a treatment. Yes, you are allowed to ask people questions. That's not imposing a treatment on them. That all falls under observational study. The one exception is an experiment. Deliberately imposes treatment on individuals to measure responses. So right away, an experiment is nothing where you can look at past data, because that's stuff that just already happened. So an experiment is kind of something that's going on. You have to impose a treatment and measure responses. So when our goal is to understand causing and effect, cause and effect, experiments are the only source of fully convincing data. So if you really want to look at something causing something else and you want to be able to limit different sort of surrounding variables, the experiment is the really only source for convincing full-on data. The distinction between observational study and experiment is one that is most important. So before we continue, make sure you understand that when you're writing about observational studies and you're writing about experiments, things aren't always exactly written the exact same way either. So be careful with your wording, and let's see why we do an experiment. Observational studies of the effect of an explanatory variable on a response variable often fail. Meaning, what does that mean? Cause and effect. They often fail because of confounding. The word is confounding, another vocab word in a very vocab-heavy chapter, between the explanatory variable and one or more of the responses. Confounding occurs when two variables are associated in such a way that their effects on the response variable cannot be distinguished from one another. Easiest one I always like to think about is they say that red wine, that when your people are of age can drink red wine, they say, oh, it's good for your health. But there's also the same exact people who drink one glass of red wine who typically have overall better diets. That's another proven fact. So is it the red wine, a glass a day of red wine, helping? Or is it that those people typically are linked to already having better overall diets? That's the idea of confounding. So well-designed experiments take steps to prevent that. So in terms of saying, oh, I have these things, but it could be many different variables affecting it, in a well-designed experiment, you try to limit confounding, we call it, limit. So here it is. I said there's some important language in experiments. An experiment is a statistical study in which we actually do something. It's called a treatment. You impose a treatment on people, animals, objects. We call those the experimental units. Okay, also called the subjects. The people, the animals, the objects are all experimental units. But when it is people, we call them subjects. Here's basic vocabulary. A specific condition applied to the individuals in an experiment is called the treatment. If an experiment has several explanatory variables, a treatment is a combination of the specific values of those variables. The experimental units are whatever objects you are collecting. They are the individuals you are collecting data about. And if they are humans, we call them human subjects in an experiment. Now, just like we did last section, let's start with the bad. Many laboratory experiments use a design like the one in our SAT course example from your reading. What did they do in the SAT course of example on the reading? They said, hey, let's get experimental units. Let's impose a treatment and let's see what the response is. Did they do better on the SAT? Why is this experimenting badly? Because this is called simple design, right? This is just simple design. In a lab environment, simple design might work well. But in field experiments, meaning where you can't really control everything with animals or even, and mostly people, there are way more variables at work. So outside the lab, badly designed experiments, when you just do a simple 1, 2, 3 like this, typically don't work. What is missing here? What would turn this into a better design? A control group. From your reading, we talked about experiments having 2 groups. So how to experiment well is to put the idea of comparison into your experiment. The remedy for confounding is to perform a comparative experiment in which some units receive one treatment and similar units receive another. Most well-designed experiments compare 2 or more treatments. Comparison alone isn't enough. If the treatment are given to the 2 groups that differ too much, we will get bias. How do we fix bias? We learned that last section. Randomize. So not only do we need to have comparison, we also need to have randomization in a good experiment. So in an experiment, we use random assignment of the experimental units to their treatment groups. See, last section, we talked about simple random sampling. Here we talk about random assignment of treatment groups. See, the wording gets a little touchy here. So, principles of our basic experimental design. 1, you need to have some sort of comparison of some sort. So you need to basically have at least 2 treatment groups. One might be getting something called a placebo, if it's a medication or something. One group might just be called the control group, meaning they try and just keep the variables the same, except the treatment. So you have to have comparison. After you have comparison, you have to randomly assign your subjects to treatment groups. So this is still, we just learned last chapter how to use the table of random digits. We just learned last section how to use randint and randint no rep on the calculator. If the table of random digits, randint or randint no rep, are some things you can't do, you need to revisit section 1. Okay, so you need to randomly assign your treatment, your experimental subjects to their treatment groups. You have to have what's called the control. I said in a comparison, you usually have a treatment group and a control group. What happens in the control group? You basically keep all of the variables as constant as possible, except the one part of the experiment, the treatment you are trying to impose. You try and keep that different, and then you replicate. Use enough experimental units in each group so that any differences in the effects of the treatments can be distinguished from one chance difference between the groups. So, a good experiment has comparison, randomization, a group that is called the control, and then you repeat with as many people or as many times as you can do the experiment. This is our typical. We had SRS in simple random sampling. In an experiment, we have the completely randomized design. It's the most basic of the designs. The treatment are assigned to all experimental units completely by chance. Some experiments may include the control group. So basically, we have a treatment group and a control group in our basic. Here's what it looks like. Okay, you're going to be drawing these pictures. Get an idea of what this looks like. So what it would be here, you wouldn't use these basic terms. You would explain to me what's happening here. But this is the framework for a completely randomized design. It's just this simple. You would say here how many experimental units there are and what they are. Let's say this could be 100 people. This could be 14 fire engines. Whatever it is, you write what is the total here. Then you do an arrow. Then you say random assignment. Sometimes they will ask you to describe how you randomly allocated people. You could say, explain how you numbered them 0, 1 through 14. You use the random digit table, and you assigned 7 people to group 1 and 7 people to group 2. You could say you went to the calculator and did randint no rep 1, 14. And the first 7 numbers that came up, were your group 1. The rest of the numbers were group 2. But sometimes they will not require you to explain that. The questions will be very specific if it says described how you randomly assign treatments. If it doesn't, you could just write random assignment. Just that simple. Group 1, you will tell me here, did this turn into 50 people? Was this 7 fire engines? Group 2, was this 49 people? 6 fire engines, 7 fire engines, because these groups don't have to be equal. Then you tell me what treatment this gets. Maybe this treatment was fire engine painted yellow. Maybe this treatment was fire engine remained red. And then you compare results. Well, maybe they were wondering, does a yellow painted fire truck get into less accidents than a red one? So you would compare the results over time on, did any of these fire engines get into accident? And that would be an experiment. This is what the basic completely randomized design looks like. You would write in all of these boxes specifics for that experiment. What can go wrong with experiments? We looked last section at some very important things that can go wrong with sampling. Well, this happens with our experiments also. The logic of a randomized comparative experiment depends on our ability to treat all of the subjects in the same in every way except for the actual treatment. Good experiments, therefore, require careful attention to detail. So we have two different things that we try and look at when we look at experiments. The first one is the idea of what's called the placebo effect. The idea of what a placebo is very much related to drug testing. When someone has, for instance, a headache, and you say, take an Advil, there is something that is well documented called the placebo effect, where if you took anything for that headache, your headache might go away. Meaning I could have given you a fake pill of sugar, and that would have been a placebo, no actual painkiller in it. And your headache could have gone away. So the idea here is you have to look out when you make an experiment for what's called the placebo effect, meaning just getting anything could cause a response. So basically, what does this mean? How does this work in the drug industry? It means if one group's getting Advil, the other group gets something that is identical looking to Advil. But who knows what they're getting? Nobody. It's called blinding. In a blind experiment, it's hidden from people what group they're in. Basically, one group might be getting Advil, one group might be getting sugar pills. Blinding means don't tell them. Double blinding means don't tell the subjects and also don't tell the person who's giving them the pill. That way they treat everyone the same. The person who's handing you the Advil or fake Advil doesn't even know if it's Advil or fake Advil. That way nobody in contact with the subjects ever even knows. And that is trying to, again, limit bias, limit confounding variables. That's what can go wrong with an experiment, basically. The inability to limit the confounding variables, not blinding, and you always have to look out for the placebo effect, just having an effect from something fake. So inference for experiments. In an experiment, researchers usually hope to see a difference in the responses, so large that it's unlikely due to chance. Meaning, if one group got headache medicine and one group got sugar pills, and when they get headaches, the group that got the headache medicine better have statistically significantly had better results. We can use the laws of probability, which describe chance behavior, to learn whether treatment effects are large enough to say it's not just chance. So we get a vocab word called statistically significant. In a chapter full of vocab, we have more. An observed effect so large it would rarely be chance alone is called statistically significant. Statistically significant association in data from a well-designed experiment is how you can apply cause and effect, causation. This is very important. It's very hard to look for something causing something else in a random sampling. And in an experiment, we need to have the data be so evident we can call it statistically significant. Meaning if one group had 49% and the other group had 50% get better, there's no way that that is statistically significant. It has to be wide results, very pronounced. We'll add numbers to this later. Okay, just like we had stratified random sampling after simple random sampling, we have the exact same thing as stratified for experiments, but it's called blocking. So you cannot call it a stratified experiment. This is called a blocked experiment. This is blocking, but it's the same exact thing. Look here at this design. When a population consists of groups of individuals that are similar within, but different between, you probably want to stratify them. So this was interesting one. This was from the reading where it said, we wanted to test a new keyboard on people, but we really think that smartphone users will use the keyboards differently than people who are still on an old-fashioned flip phone. So they said, we're going to block. And look what happens with blocking. You still start with the same group, one group of 10 volunteers. Then you, on purpose, move them into the smartphone group and the non-smartphone flip phone group. Then look what happens. You have to randomly assign after the blocking so that the people who are supposed to be in this group are in this group. You randomly assign them. They did 2 people and 2 people here to the new keyboard and a different keyboard. They looked at results, and then they also did the same exact thing. They did basically a simple experiment, right? That randomized design for each of these groups. So 2 separate experiments, essentially. Non-smartphone users got randomly assigned to 3 people here, 3 people here. The treatment was the keyboard and the other keyboard. They compared results. And then finally, you can finally go back and compare results to the original 10 people. And you can look into each block's results separately. It's the same as stratified, where you do something on purpose, but then you do multiple randomized experiments after that. This can be way more than just 2. You can have 1, 2, 3, 4 groups, 5 groups, whatever it takes. But at first it's on purpose, and then you still use randomization after. We also could have multiple treatments, right? You could have keyboard A, keyboard B, keyboard C. The sky's the limit. So a block is a group of experimental units that are known before the experiment to be similar in a way that you want to separate them. In a randomized block design, the random assignment of experimental units to the treatment groups is carried out separately, right here, after you already switch and separate the blocks. Our last type is a little bit different. It's a special type of blocking, a common type of randomized block. So it's a special block for comparing 2 treatments is called a matched pairs design. The idea here is to create blocks by matching pairs of similar experimental units. So a matched pairs design is a randomized block design in which each block consists of a matched pair of similar units. Chance is used to determine which unit in each pair gets which treatment. So what does that mean? It means, look at me, okay? Look at me. I am a middle-aged, 5'5" male. Sometimes it helps if in an experiment I was matched up on purpose with a similar middle-aged, give or take 5'5" male, because that limits a lot of variables. It's trying to limit confounding. It's the big important part of an experiment. Try to limit as many differences as possible, and a matched pairs as possible like that. So on purpose, you match me with someone very similar to me. Then we randomly assign which treatment each of us gets, and we compare mine and the other person's results as part of the bigger experimental conclusion. So I was matched up with someone. The last part about this is I could be my own pair. Meaning, if you wanted to see how well I typed on keyboard A and keyboard B from here, myself as one person, I might be a matched pair with myself. And potentially, I might be told, randomize, do I work on keyboard A first or do I work on keyboard B first? That would be my random assignment. So it's two types of really matched pairs, but they're both matched pairs. It's a block design where you match one person to another, or where one person gets both sides of the experiment and you randomly assign which one they get first. This is, again, a special type of a blocked experiment. So we, again, have a completely randomized design. That's our basic, simple version of experiment. Then we have a block design, and then a special block design is the matched pairs design. And that was it for experiments. A lot of new language here again.